Let's start with something you already know about growth. Plants grow bigger, trees get taller, leaves get larger. But what exactly is happening? Growth is defined as an irreversible, permanent increase in size. Notice I said irreversible means once something grows, it can't go backward to being smaller. This could be the whole organ growing, just part of it, or even individual cells getting larger. Here's the important part. Growth doesn't happen by magic. It requires energy. Your body uses energy and so do plants. This energy comes from metabolic processes, the chemistry happening inside cells. Some of those processes build things up, others break things down, but all of it requires energy expenditure. Now let me give you a concrete example so you understand the difference. When a leaf expands, that's growth. It's permanent. But here's something tricky. If you take a piece of wood and soak it in water, it swells up, right? That looks like growth, but it's not. Why? Because it's temporary and reversible. The moment you dry that wood out, it shrinks back to its original size. That's not growth. Growth has to be permanent and irreversible. This distinction is important when we're measuring and studying plant development. Here's something really fascinating about plants. They never stop growing. Your body stopped growing at some point, right? But a tree can keep growing for decades, even centuries. Why? Because plants have something animals don't have. Meristems, these are special regions in plants where cells have a remarkable ability. They can divide and they can keep on dividing forever. These cells self-perpetuate, meaning they continually create new cells. It's like having a fountain that never runs out of water. As long as the plant is alive, meristems are producing new cells. This is called the open form of growth, an open-ended process with no built-in stopping point. Let me break down how this actually works. There are two types of growth happening. First, primary growth. This happens at the tips of roots and shoots. We call these the apical meristems. This is what makes plants grow taller and longer. The root extends downward, the shoot extends upward. But there's more. In trees and other plants, secondary growth happens through lateral meristems, specialized areas on the sides called the vascular cambium and cork cambium. This secondary growth is what makes trees get thicker and wider as they age. So a 50-year-old oak tree has been doing both, growing taller for a while, then getting progressively thicker year after year. So how do scientists actually measure growth? It depends on what's being measured. You could weigh something fresh weight or dry weight. You could measure how long it is. You could measure its surface area or volume. You could even count the number of cells. The key is choosing the right measurement for what you're studying. Different organisms grow in different ways, so different measurement techniques make sense for different situations. Let me give you some amazing numbers. One single maize root tip, just one, meristem produces more than 17,500 new cells every single hour. That's incredible. But here's something else incredible. Watermelon cells can grow. A single watermelon cell can increase in size by up to 350,000 times. Compare these two. The maize root is showing growth through cell division, lots and lots of new cells. The watermelon is showing growth through cell expansion, means each individual cell getting enormous. Both are growth, but they're happening through completely different mechanisms. This is why scientists have to think carefully about measurement. With a pollen tube, which is really a long, thin structure, length makes perfect sense to measure. But for a leaf, especially a leaf that's flat and wide, measuring the increase in surface area tells you much more useful information than length would. The measurement has to match what you're studying. Let's follow a plant cell's journey. The first phase is the merismatic phase. Imagine the very tip of a root or shoot, that's where you'll find merismatic cells. These cells are dividing constantly, relentlessly. If you look at them under a microscope, they're packed with protoplasm, means that's the living material inside the cell. The nuclei are huge and very obvious. These are young, active, undifferentiated cells. What about their cell walls? Their primary cell walls, meaning their first, original cell walls, they're thin and made of cellulose. There's something else important. These cells are connected to each other by plasmodesmata, fissure. These are like communication tunnels between cells. There are lots of them, creating a network that lets cells talk to each other. Next comes the elongation phase. This zone is right next to the merismatic zone. These cells aren't dividing as much anymore, instead they're expanding. What's happening? Vacuoles are forming and getting bigger. Those are the water-filled spaces inside cells. The cell walls are getting thicker. The whole cell is getting larger. This is where most of the actual lengthening of the root happens. Finally comes maturation. This is further away from the growing tip. By now, the cells have reached their full size. Their walls have thickened completely. The protoplasm has undergone all its modifications. The cells have specialized for whatever job they're going to do. When you look at plant tissues under a microscope in a typical textbook, you're usually looking at cells in this maturation phase. They've finished their journey from simple dividing cells to specialized functional cells. Now, let's talk about growth patterns. There are two main patterns plants follow. The first is reversible growth. Here's how it works. A cell divides. Two daughter cells are produced. But then something happens. Only one of them keeps dividing. The other one stops dividing and specializes into whatever cell it's going to become. This pattern continues, so you get a steady, constant rate of growth. The best example of irreversible growth is a root growing. It elongates at a constant rate. If you drew a graph with time on one axis and length on the other, you'd get a straight line, a perfect linear relationship. And that straight line can be expressed with a simple mathematical formula, L sub t equals L sub 0 plus R times t. Let me break this down. L sub t is the length at any time you're measuring. L sub 0 is the starting length at the beginning. R is the growth rate, how much it grows per unit of time. T is the time. So it's a simple equation describing that straight line growth. Most real-world growth doesn't follow the simple arithmetic pattern. In geometrical growth, which is what we usually see in nature, there are three distinct phases. First, the lag phase. Growth starts slowly. Things are getting set up. Then comes the exponential phase. Suddenly growth explodes. Everything's in place. Resources are flowing. Cells are multiplying rapidly. But eventually, the resources run out. Nutrients become limited. Growth slows down. That's the stationary phase. Here's the key difference from arithmetic growth. When a cell divides in geometrical growth, both daughter cells keep the ability to divide. They don't stop and differentiate. They keep on dividing. That's why growth accelerates so dramatically. It's exponential, meaning it keeps speeding up. Here's the magic part. If you graph this geometrical growth, plotting the growth parameter on one axis and time on the other, you get a beautiful S-shaped curve called a sigmoid curve. It starts flat during lag phase, shoots up steeply during exponential phase, then levels off during stationary phase. This S-curve is the signature pattern of living organisms growing in nature. You'll see it in cells, in tissues, in organs. It's universal. The mathematics looks more complicated, but it's actually elegant. W sub 1 equals W sub 0 times e to the power of RT. W sub 1 is your final size, height whatever you're measuring. W sub 0 is where you started. R is the relative growth rate, thickens. This is also called the efficiency index because it measures how efficiently the organism produces new material. T is time and E is the mathematical constant, the base of natural logarithms. The beautiful thing about this formula, it shows that the final size depends on both how fast you grow and how much you started with. Sometimes you need to compare growth between different organisms or structures. There are two main ways to do this. First, absolute growth rate. This is just the total amount of growth in a unit of time. For example, if something gained 10 grams in 1 week, that's the absolute growth rate. It's straightforward. If you're comparing two similar organisms, absolute rates work well. But here's the problem. What if you're comparing a large organism with a small one? A large tree might add more total mass than a small seedling, but the seedling is actually growing faster in relative terms. That's why we use relative growth rate. You express growth as a proportion of the starting size. A small organism that doubles in size has a higher relative growth rate than a large organism that gains the same absolute amount of mass. For fair comparison between different sized organisms, relative growth rate is the right tool. What does a plant absolutely need to grow? Water is at the top of the list. Plant cells grow by getting bigger by expanding. That expansion requires water. It's the pressure of water inside cells' rigidity that pushes outward and helps the cell walls extend. Without adequate water, cells can't expand, and without expansion, most growth can't happen. It's that fundamental. But there's more to water's role. All those chemical reactions happening inside cells, the metabolism, they happen in water. Water is the medium where enzymes work. So plant growth and development are deeply tied to how much water the plant has available. A drought-stressed plant won't grow even if it has all the nutrients it needs because it doesn't have enough water for its enzymes to work. Next is oxygen. Oxygen is what allows cells to release the energy stored in food molecules. Through respiration, plants break down glucose and other molecules, and oxygen helps them capture that energy in the form of ATP. That ATP then powers all the work of growth. No oxygen means no energy release, means no growth. Then there are nutrients, both macronutrients and micronutrients. These are the elements nitrogen, phosphorus, potassium, calcium, magnesium, and all the trace elements. Where do they go? They are incorporated into protoplasm, their living material of cells. They are not just consumed, they become part of the cell structure itself. Plus, some nutrients are used as energy sources. Temperature matters too. Every plant has an optimal temperature range. They range where it grows best. Too cold and enzyme reactions slow down almost to nothing. Too hot and proteins denature. The sweet spot varies by species. Tropical plants have different optimal temperatures than Arctic plants. Even small deviations from optimal can be harmful. And don't forget environmental signals. Light affects development. Plants sense light and respond to it. Gravity too. Roots know to grow downward. Shoots know to grow upward. These environmental factors interact with the internal chemical signals in plants to direct growth. So far we've talked about growth cells dividing and expanding. But something equally important happens, differentiation. This is when cells stop being general purpose cells and become specialized. A cell produced by a meristem might initially be pretty generic, but then it receives signals, positional signals, chemical signals, that tell it what job to do. It transforms into a specialized cell designed for that specific function. During differentiation, cells undergo dramatic changes. Their cell walls change, they might become thicker or thinner or develop special properties. The protoplasm inside changes too. Some structures are lost because they are no longer needed. New structures are gained because they are essential for the cell's new job. Let me give you a dramatic example. Physiology. These are the water-carrying cells in plants. When a cell differentiates to become a xylem vessel, it undergoes an extreme transformation. It loses its protoplasm, lymph dies. What's left is a hollow tube. But this tube has incredibly strong, thick walls made of lignin and cellulose. These walls can withstand the extreme tension of water being pulled up a 100-meter tall tree. It's a perfect structure for its function carrying water. A generic cell could never do this job. Only a specialized xylem vessel can. Here's something remarkable. Cells that have already specialized can sometimes go backward. They can de-differentiate, lose their specialization, and regain the ability to divide. This is amazing when you think about it. A cell that had decided what it was going to be can change its mind under the right conditions. Here are real examples. In plants with secondary growth, something special happens. Parenchyma cells go somnigized. These are pretty generic living cells. Can they differentiate and become interfascicular cambium or cork cambium? These are meristematic tissues that keep dividing for years. The cells went from being differentiated, non-dividing parenchyma to dividing meristematic cells. Then redifferentiation happens. The newly formed cells from these meristems go through the whole process again. They divide, then specialize into whatever tissue is needed. This back and forth differentiation to de-differentiation to redifferentiation shows that plant cells are incredibly flexible. This flexibility is actually what allows plant tissue culture to work. Scientists take differentiated cells, de-differentiate them in culture, and then redifferentiate them into new tissues or whole new plants. Here's a key insight. Differentiation in plants is what we call open, cells that come from the same meristem, the same source, end up being completely different cells. Why? Because of where they are. Position matters enormously in plant development. The same genetic code is present in all these cells, but their location tells them who they should become. Let me show you what I mean. At the root tip, there's a root apical meristem creating new cells. Those cells that end up positioned away from the apex pushed backward. They become root cap cells. They protect the root as it pushes through soil. But those cells that get pushed toward the periphery, to the outer surface, they become epidermal cells. Same meristem, same time period, different positions, completely different cell types. Position determines fate. Now, we are ready for the big picture. Development is everything. It's the sum of all the changes a plant goes through. It starts with seed germination, that first crack in the seed coat. It continues as the plant grows and differentiates. It includes flowering, fruiting, seed production, and eventually, it includes senescence, aging, and death. All of that is development. And development is made of two processes, growth and differentiation, working together. Plants are incredibly flexible, more so than you might think. This flexibility is called plasticity. It's the ability to respond to your environment by changing your developmental pathway. Different conditions trigger different outcomes. Same genetics, different environment, completely different structures. Here are examples. Look at cotton plants or coriander or larkspur. When the plant is young, it makes certain shaped leaves, but as the plant matures, it makes completely different shaped leaves. Same plant, different life stage, different structure. The plant decides when to switch. But there's even more remarkable plasticity based on environment. Look at buttercup plants that live in water or partially in water. The leaves that develop in air have one shape. The leaves that develop underwater have a completely different shape. The plant senses whether it's in water or air and adjusts its leaf development accordingly. It's not random, it's a specific adaptation to the environment. Both of these phenomena, changing shape with age or environment, are examples of heterophyly, which is plasticity in action. It's the plant saying, I have the genetic potential to make different leaf shapes and I'll choose which one based on where I am or how old I am. It's beautiful adaptation at the developmental level. To make a stance. So what controls all this development, growth and differentiation? There are two levels of control. First, intrinsic factor, things happening inside the plant. Your genes are intrinsic, but genes alone don't determine development. There's also chemistry. Cells produce chemical signals, plant growth regulators, hormones that tell cells what to do. These chemical messengers coordinate development throughout the plant body. But plants aren't isolated from their environment. Extrinsic factors, external factors matter greatly. Light, temperature, water, oxygen, nutrients in the soil. The environment sends signals. A plant doesn't develop the same way in full sun as it does in shade. It doesn't grow the same in hot weather as in cold. Development responds to these external conditions. Now we get to the chemical messengers inside plants, the plant growth regulators or PGRs. Sometimes you'll hear them called plant hormones or phytohormones. These are small molecules, chemically diverse, but all of them have big effects on how plants grow and develop. What's remarkable is how little you need. Tiny concentrations of these molecules can completely change what a plant does. Let me introduce the 5 main groups. There are indole compounds. These are the auxins, and IAA is the most famous one. There are adenine derivatives, the cytokinins, which include kinetin. There are carotenoid derivatives. This is where abscisic acid comes from. Then there are terpenes. This class includes gibberellic acid or GA3. And there's ethylene, the only gaseous PGR, a simple gas. These 5 groups cover most of the major growth regulation in plants. Here's a useful way to think about PGRs. One group are growth promoters. These speed things up. Auxins, gibberellins, and cytokinins all push plants to grow and develop. They promote cell division, cell enlargement. They help pattern formation during development. They drive flowering, fruiting, seed production. They control the directional growth toward light or against gravity. The other group are growth inhibitors. Abscisic acid is the main one. These PGRs do the opposite meaning. They slow things down or stop them. They're especially important when plants face stress, drought, injury, disease. They trigger dormancy to conserve resources. They cause leaves and fruits to drop. When conditions are tough, they're like the plant's safety system, helping it survive hard times. Then there's ethylene, the odd one out. It's unique. Because it's a gas, so it behaves differently. It can fit into either category. It primarily inhibits growth, but it has some promoting effects too. It's like the bridge between the two groups, showing that growth regulation isn't always simple on-off switches. Let me tell you a detective story of scientific discovery. It starts with Charles Darwin and his son Francis. They were studying young grass shoots called aptiles, and they noticed something. When light came from one side, the shoots bent toward it. They called this phototropism, growth, directed by light. This observation puzzled them because the bending happened below where the light was hitting. Here's what puzzled them. The light hit the very tip of the shoot, but the shoot bent much lower down. How does the tip tell the rest of the shoot to bend? They concluded there must be some transmittable influence, some signal traveling from the tip down to the region that bends. Then came F.W. Went in the 1920s. He had a brilliant idea. He took up tips and put them on agar, gel-like substance. The tip cells secreted something into the agar. When he took that agar block and placed it off to the side of a shoot without any light, the shoot bent anyway. This proved it wasn't light doing the bending, it was a chemical that had diffused into the agar from the tip. That chemical was later identified as indole 3-acetic acid, or IAA, the first auxin ever identified. Here's another discovery story. Japanese farmers noticed something strange in their rice paddies. Some seedlings were growing abnormally tall and spindly. They called it bakmane, foolish seedling disease. The disease was caused by a fungus called Gibberella fujikuroi. Scientists were intrigued. Why did infection make plants grow so tall? In 1926, E. Kurosawa did something clever. He filtered the fungal culture so that no living fungus remained, just the liquid where the fungus had grown. He applied this sterile filtrate to healthy rice seedlings, and amazingly, the seedlings developed the same symptoms. This meant a chemical, not the living fungus itself, was causing the effect. They isolated the chemical and identified it. They called it gibberellic acid, or Ga3. Since then, scientists have found over 100 different gibberellins in nature. They are produced by fungi and higher plants. The fungus that causes rice disease was accidentally producing a plant hormone that happens to stimulate growth. That accidental discovery led to one of our most useful plant hormones. Here's another important story. F. Skoog was working with tobacco plant tissue in the laboratory. He was trying to grow callus pip. That's an undifferentiated mass of cells from tobacco stem tissue. He knew auxins were needed, but something else was also required. In addition to auxins, he needed to add something else to the growth medium. When he added plant tissue extracts, it worked. When he added yeast extract, it worked. When he added coconut milk, it worked. When he even added DNA, it worked. Something in all these different sources was promoting cell division. What could be common to all of them? In 1955, Miller and his team took this detective work further. They isolated and crystallized the active substance. They called it kinetin, a modified form of adenine, one of the bases in DNA. Kinetin doesn't occur naturally in plants. It was a laboratory creation, but it led scientists to search for natural versions. They found zeatin in corn kernels and coconut milk. So that's why coconut milk worked in Skoog's experiments. It contained natural cytokinins. In the mid-1960s, something interesting happened. 3 different research teams, working independently, each discovered what they thought was a new growth inhibitor. One team called theirs inhibitor B, another team called theirs epsilon Pud, because it seemed to cause leaves and fruits to drop off. The third team called theirs domin, because it seemed to cause dormancy. But then when scientists analyzed them chemically, they discovered they were all the same molecule. 3 separate discoveries of the same hormone. They merged the names and called it abscisic acid ABA, keeping both the epsilon and the sense of inhibition implied in all 3 original names. Synchronicity successlati nautipore. And ethylene back in 1910, H. Cousins made an observation in a fruit storage facility. Ripe oranges were being stored near unripe bananas. The bananas ripened much faster than expected. He realized the ripened oranges must be releasing something that affected the bananas. He identified the substance as ethylene, with pimple gas, just 2 carbon atoms and 4 hydrogen atoms. It's the only plant hormone that's a gas. The word auxin comes from Greek. Xen means to grow. It was first identified in human urine, which is kind of interesting when you think about it. Now, we use auxin as a general term for a class of growth regulators with similar effects. Auxins are small molecules that regulate growth. They're made in the growing tips of roots and shoots. The interesting thing is that they're not used where they're made. They migrate. The tip produces the auxin, then it travels down the shoot to where it does its job. And importantly, the effect depends on concentration. High concentration causes one effect, low concentration causes another. Let me distinguish between natural and synthetic forms. In nature, plants produce IAA, indole thioacetic acid hydro. This is the main one. There's also IBA, indole 3-butyric acid, which occurs naturally too. Scientists have also created synthetic auxins that have similar effects. NAA and 2,4-D are synthetic. 2,4-D is especially famous because it's used as a selective herbicide. All these auxins, natural and synthetic nuts, have been used extensively in farming and gardening because they're so practical. What do auxins actually do? First, they promote rooting. If you take a cutting from a plant and want it to develop roots, auxin is your answer. Gardeners and horticulturists use auxin to dip cuttings in before planting them. The auxin stimulates the formation of adventitious roots, roots that develop from the cut stem. This is how many plants are propagated commercially. Auxins also promote flowering in some plants. Pineapple farmers use auxin to trigger flowering at the right time. Instead of waiting for the plant's natural flowering time, they can use auxin to synchronize flowering across their crop, making harvest more efficient. Here's something elegant. The same hormone auxin can prevent drop or promote drop depending on when it's used. Early in the season, you want to keep fruit and leaves on the plant, so auxin helps prevent ipsation. Later, when fruit is mature and ready to harvest, or when leaves should drop, you can manipulate auxin levels to promote ipsation. It's all about concentration and timing. Auxins also induce parthenocarpy, the development of fruit without fertilization. In tomatoes, this creates seedless fruits. Normally, a fruit develops after fertilization and contains seeds. But with auxin, fruit can develop without any seeds. Consumers like seedless fruits, so this is commercially important. Have you ever noticed that a tree or plant tends to grow one main stem instead of branching early? That's apical dominance. The growing tip produces high concentrations of auxin that suppress the development of side branches. It's a growth strategy focus energy on reaching upward toward the light rather than spreading out sideways. Here's the proof of the mechanism. Remove the growing tip, decapitate the plant. Without the tip producing auxin, those suppressed lateral buds suddenly wake up and start growing into branches. This simple experiment proves that auxin from the apical meristem is what's suppressing the side branches. This knowledge is used practically. Tea farmers regularly remove shoot tips from tea plants to encourage them to become bushy rather than growing as single tall stems. This creates more harvestable tea-producing branches at a convenient height. Hedge makers do the same thing. They prune the tops to create fuller, bushier hedges. It's an elegant application of plant physiology. Here's a completely different use of auxins as herbicides. At high concentrations, auxins actually kill plants by disrupting their growth patterns so severely that the plant cannot survive. It's like giving the plant an overdose. 2,4-D is particularly useful because it's selective. It kills dicots plants with 2 cotyledons, like most broad-leaved plants, but it doesn't hurt mature monocots plants with 1 cotyledon, like grasses. This selectivity is crucial. So what does a gardener do? Most lawn grasses are monocots. Most weeds invading lawns are dicots, things like dandelions. A gardener sprays the lawn with 2,4-D. The dandelions die. The grass lives. It's weed-free lawn without having to pull every single weed by hand. Beautiful application of plant science. Let me tell you what's amazing about gibberellins. There aren't just one or two. Why? Over 100 different gibberellins identified in nature. Scientists have found them in fungi, in higher plants, even in algae. They're incredibly diverse. GA3, gibberellic acid, was one of the first ones found, and it's still the one scientists study most. It's the most commercially useful. No matter which gibberellin you're looking at, they're all acetic wine. It's a constant chemical feature. What do gibberellins do? That's the interesting part. They do many things. They have a wide range of physiological effects. The specific effect depends on what tissue you're looking at and what stage the plant is in. Gibberellins are like orchestral conductors. They regulate many different processes.
Let me give you practical examples. In grape farming, they use gibberellins to increase the length of grape stalks, the stems that hold the individual grapes. Longer stalks make the grape clusters look better and more appealing to consumers.
Gibberellins also affect the shape of fruits. They cause fruits like apples to elongate and improve their overall form. Better looking fruit commands higher prices. Here's something economically important. Gibberellins delay senescence. That's the aging process. So a fruit stays fresher on the tree longer than normal. Farmers can harvest later, giving them flexibility to sell when prices are better. It extends the market period for that crop. And here's an industrial use. GA3 is used in brewing. The malting process, converting grain to malt, normally takes days. Gibberellin speeds it up. It's all about those enzymatic reactions happening faster. Sugarcane is an interesting crop. The plant stores its sugar in the stem. So if you can make the stem longer, you can store more sugar, which means more product to harvest. That's the key insight here. When farmers spray gibberellins on sugarcane, the stems grow longer. This isn't just a little improvement. We're talking about increasing yield by up to 20 tons per acre. That's massive. For a farmer, that's the difference between a good year and a great year. Here's a different application. Forestry. Conifer tree spines, firs, normally take years to mature and produce seeds. When foresters spray juvenile conifers with gibberellins, it speeds up maturation. The trees flower and produce seeds earlier. This is valuable for breeding programs and for forestry management. Let's talk about bolting. Because this is when a plant suddenly grows rapidly tall, with long spaces between leaves. This happens just before flowering. The plant is shifting from vegetative growth, making leaves and stems, to reproductive growth, making flowers and seeds. Gibberellins are what trigger bolting. In plants like beet or cabbage that have a rosette habit, their leaves are arranged in a circle at the base. Initially, gibberellins cause the stem to suddenly elongate and produce the flower stalk. Some plants are biennial, so bolting happens in the second year. Cytokinins come from a different discovery path. Remember Cook's tobacco experiments? That mysterious factor in coconut milk and yeast extract that promoted cell division? Scientists isolated it and identified it. The first one they found was kinetin, a modified version of adenine, one of the DNA bases. Interestingly, they first found it in autoclaved herring sperm DNA, named pretty random source. Here's something important. Kinetin doesn't naturally occur in plants. It's a laboratory creation. But because scientists found it had cell division promoting activity, they realized plants must produce something similar. That's when they found zeatin, the natural version, in corn kernels and coconut milk. Suddenly, Cook's results made sense. Coconut milk had natural cytokinins in it. Since then, scientists have identified multiple naturally occurring cytokinins and created various synthetic forms with similar activity. Where are cytokinins made in plants? Wherever cells are dividing rapidly. That makes sense, right? Cytokinins promote cell division, so they're made where cell division is happening. The root tips are one place, lots of cell division happening there. The developing buds, the belly, is lots of division. Young developing fruits, lots of cell activity. What do cytokinins do? First, they promote the production of new leaves. They stimulate the formation of chloroplasts inside leaves, the organelles that do photosynthesis. They promote lateral shoots, branches growing from the sides. They can even cause shoots to form in unusual places and adventitious locations. Basically, Wherever there's growth and development, cytokinins is involved. Remember apical dominance? Auxins suppress lateral buds. Cytokinins do the opposite, they promote lateral bud growth. They help overcome apical dominance. This is a beautiful example of how different hormones can have opposing effects, creating a balance that gives plants flexibility. Finally, cytokinins delay aging. How? By promoting nutrient mobilization, moving nutrients from old leaves to new growth. Instead of the old leaf dying, those nutrients are recycled to new parts. So old leaves stay functional longer, stay green longer. It's like the plant is renewing itself. Ethylene is weird compared to other plant hormones. It's a gas, just 2 carbon atoms and 4 hydrogen atoms, incredibly simple. But that gaseous nature changes everything about how it works in plants. It can diffuse through air, it can escape from cells, it travels differently than liquid hormones. Where is ethylene made? Mainly in cells that are aging or ripening. As a fruit ripens, it produces more and more ethylene. It's like the ripening process feeds back on itself. The more a fruit ripens, the more ethylene it makes, which causes it to ripen faster. Let's see what ethylene does. In seedlings, young plants just sprouted, ethylene causes some interesting changes. Instead of growing straight up, seedlings tend to grow horizontally. The stem swells up. In dicots plants with 2 cotyledons, a little hook forms at the tip. This hook helps the seedling push through soil. The most famous role of ethylene is in fruit ripening. Ethylene is incredibly effective. A tiny amount triggers the whole ripening process. It increases the respiration rate dramatically. That surge in metabolic activity, that increased breathing, is called the respiratory climacteric. It's the metabolic switch that flips ripening on. Ethylene also wakes things up. Seeds that are dormant sleeping can be awakened by ethylene. In peanuts, ethylene initiates germination. In potatoes, ethylene causes the tubers to sprout. It's like ethylene is the wake-up call for seeds and buds. Ethylene also promotes root development. It stimulates the growth of tiny root hairs, those thin extensions on root cells. Root hairs greatly increase the surface area for absorption. More root hairs means a seedling can absorb more water and nutrients. It's setting the plant up for success. Here's a fascinating adaptation. Some rice plants grow in areas that flood. When water rises, the plants face a problem. They need their leaves above water to photosynthesize. What do? Ethylene steps in. It triggers rapid elongation of the internodes, rows, the stem sections, so the plant literally grows upward faster than the water rises. The internodes stretch out, the petioles leaves also elongate. The whole upper part of the plant shoots upward, staying above the floodwaters. This is ethylene-mediated survival. It's an adaptation that's been refined over many generations of rice cultivation. Pineapple plants normally flower at different times, which makes harvesting difficult. Farmers use ethylene to make them all flower at the same time. This synchronizes fruit production. Now all the pineapples ripen together, allowing efficient, coordinated harvesting. Mango farmers use ethylene similarly to induce flowering when they want it, allowing them to time production to match market demands. Because ethylene regulates so many different processes, ripening, flowering, dormancy, growth, it's become one of the most important plant hormones in agriculture. Modern farming depends on ethylene for synchronized production of so many crops. Direct ethylene gas is hard to apply to plants, it escapes. So chemists created ethylene, a chemical that releases ethylene inside the plant. It's the commercial form of ethylene used in agriculture. Ethylene is applied as a water solution. That's much easier than trying to apply gas. The solution is absorbed into the plant. It moves internally through the plant. Then, inside the plant, it slowly releases ethylene. This gradual release is actually better than a sudden blast of gassing, gives more control over the response. One use of ethylene is accelerating ripening. Tomato and apple farmers use it to ripen fruit on their schedule rather than waiting for natural ripening. When market prices are good, they can accelerate ripening and harvest at the best time. Ethylene has the opposite effect. In some cases, it causes abscission. In cotton, it causes excess flowers to drop. reducing the number of bolls so each one can grow larger and of better quality. In cherries and walnuts, it thins out excess fruit, so the remaining fruit grows bigger and better quality. Fewer, bigger, better fruits bring higher prices than many small fruits. Here's something unique. Ithethon affects which sex flowers are in cucumber plants. It promotes female flowers. Why does this matter? Female flowers develop into fruits, that's what we harvest. Male flowers only produce pollen, so promoting female flowers directly increases yield. Abscisic acids say basic ABA species ये disjoven buyers we've talked about, while auxins, gibberellins and cytokinins are mostly promoters of growth. ABA is an inhibitor. It was discovered because it seemed to cause abscission of fruit and leaf drop. But scientists found it does much more than that. It's a general growth inhibitor and it also inhibits metabolism. ABA's first major function is maintaining dormancy. It prevents seeds from germinating at the wrong time. If a seed germinated during winter or severe drought, it would die. ABA keeps it asleep, waiting for the right conditions. ABA closes the pores that let water escape from leaves. During drought, this is critical. If somda stay open, the plant will wilt and die. ABA closes them, reducing water loss, helping the plant survive. Most importantly, ABA increases stress tolerance. When plants face any kind of stress manner, whether it's disease, insects, drought, heat, salt or cold manner, ABA is activated. It orchestrates the plant's survival response. Because of this critical role in stress, it earned the name stress hormone. ABA doesn't just keep seeds dormant ants. It's involved in the whole seed development process. During maturation, ABA helps prepare the seed for the challenges it will face. By inducing dormancy, ABA protects seeds. A dormant seed can survive extreme dryness. Seeds can lose almost all their water and still survive. They can withstand temperature extremes. They can wait years if necessary until conditions are right. ABA is responsible for this incredible survival capability. Here's something important. ABA and gibberellins usually work against each other. When gibberellins are trying to promote growth, ABA is trying to inhibit it. When ABA is trying to keep a seed dormant, gibberellins would be trying to make it germinate. They're opposing forces and the balance between them determines what actually happens to the plant. Here's the reality of plant hormones. They rarely work alone. Development involves multiple PGRs interacting in complex ways. Sometimes they work together synergistically, where two hormones together have a bigger effect than either alone. Sometimes they work against each other antagonistically, creating a balance. Sometimes one hormone acts alone, but usually it's a conversation between multiple hormones. Look at dormancy when a seed is dormant. That's controlled by ABA promoting dormancy and gibberellins being suppressed. But it's more nuanced than just those two. Light, temperature, water status, they all factor in. Same with abscission, senescence, apical dominance. Each of these involves multiple PGRs negotiating what happens. That's what makes plant development so fascinating and flexible. Now here's the big picture. Plant growth regulators are important, but they're just one part of the story. There's also genomic control, your genes inherited from your parents. Your genes determine what proteins you can make, what structures you can build. PGRs are chemical signals that activate or suppress those genetic programs. They work together with your genome. And here's the elegant part. Extrinsic factors, temperature, light, all the environmental signals. They don't control development directly. They work through plant hormones. Cold triggers the water triggers of certain hormones. Light triggers the production of others. The environment speaks to the plant through the language of hormones. The hormones then tell the genes what to do. Let me give you examples of how the environment controls development through hormones. Vernalization is when a plant needs cold mite in good animal before it can flower in spring. The cold triggers hormone changes. Flowering is controlled by day length with photoperiodism. The plant measures light duration and that triggers hormonal changes. Dormancy means seasonal changes trigger hormones that shut down growth. Seed germination night, when conditions are right, hormones are activated. Plant movements toward light make that's hormones responding to light cues, all mediated by PGRs. Let's bring this together. Growth is fundamental and irreversible increase in size. We measure it many ways. It happens because of meristems that keep dividing. Plants grow indeterminately. They never stop. That growth goes through 3 phases. First, dividing cells, then expanding cells, then specialized mature cells. And growth follows patterns, sometimes linear arithmetic, more often the realistic sigmoid curve of nature. Development brings in the complexity. Cells don't just grow, they become specialized. Different cells from the same meristem become different tissues because of their position. Remarkable. These cells can even reverse specialization and start over. Plants are developmentally plastic flexible in response to their environment. Development is the result of growth and differentiation working together, creating the complex plant body. 5 groups of plant growth regulators coordinate all this. Auxins discovered through elegant experiments control roots and dominance and act as herbicides. Gibberellenses found through a rice disease who elongate stems and promote flowering. Cytokinins from tobacco tissue culture research achieve remote cell division and leaf formation. Abscisic acid discovered 3 times by accident maintains dormancy and stress tolerance, opposing gibberellins. Ethylene, the gaseous one, ripens fruit and breaks dormancy. 5 groups, dozens of specific molecules, thousands of regulatory interactions happening in every plant right now. And all of this growth, differentiation, development is controlled at multiple levels. Genes provide the instructions, hormones send the signals, environment sets the context. It's an integrated system where environment influences hormones, hormones influence gene expression creates structure and function. That's what makes plants so remarkably responsive to their world. 3 ideas to carry with you. First, development is the combination of growth and differentiation. Growth is just getting bigger. Differentiation is just becoming specialized. Development is when they work together to build a complex organism. Second, plants are fundamentally different from animals in their growth. They never stop growing. Meristems keep producing new cells. This fundamental difference explains so much about plant form and function. Remember the 5 groups and their main jobs. Auxins coordinate root formation and dominance. Gibberellins push growth and flowering. Cytokinins drive cell division and leaf formation. Abscisic acid puts the brakes on and handles stress. Ethylene ripens fruit and breaks dormancy. None of them work alone. They interact constantly, creating the sophisticated regulation that makes plants so successful.
Meet Iyal, your AI study companion. Transform PDFs into smart study guides, practice tests, and flashcards for UPSC, NEET, banking, and any competitive exam.
Master your exam preparation with Iyal.